the big problem that you're really trying to solve. How would you summarize that for people?
Trying to address microbial imbalances in bees to improve greater ecosystem health and also to support sustainable agriculture. That's the real focus of my research. We're being affected by three things, right? Pathogens, pesticides, and let's say habitat loss. We can't really address habitat loss, single-handedly, but we can address the other two. We can address pathogens and we can address pesticide exposure, or we can try to address that through developing a probiotic to improve immunity and a probiotic to detoxify pesticides from honeybees, bumblebees, carpenter bees, all sorts of different bees. They're all landing on these same flower pads. They're all sharing microbes, right? Bad microbes, good microbes. If you can help balance the microbiome or improve the good microbes in honeybees, you can potentially influence all of these other native pollinators by simply just you're giving good microbes to bees. They're getting on these flower plants, they're sharing good microbes with the rest of the pollinators, improving overall health of all pollinators, and hopefully helping to address the decline in overall insect populations.
Hey everyone, Rupesh Patel here, host of the Two Nobodies podcast, and you just heard from my guest, Brendan Daisley, who is a honeybee microbiome researcher. Yeah, that's right. There are people who research the honeybee microbiome. They're trying to understand the links between the microbiome of a honeybee and their overall health and how they can sustain themselves, throughout the winter, and just ensure that bee populations continue to succeed. And there's so many pressures on honeybees, whether it be from pests to different pathogens to pesticides to the lack of biodiversity. And Brendan and I get into all of those things. We also talk about his research into probiotics and how probiotics can be beneficial for honeybees, but also probiotics for humans. There have been many times where I've talked about probiotics for humans, and we get into how they can also be beneficial in some ways too. So really neat episode about honeybees, the microbiome, and if you're a beekeeper or someone who's just generally interested in saving the bees, this is a really cool conversation that I think you're really going to enjoy. Cheers. Thanks.
Welcome to the Two Nobodies podcast with my dad.
Hey Brendan, good to see you. It's this has been something that I've been looking forward to. We're going to be talking about bees today. We're setting this up for World Bee Day. This is a really cool topic. Your Emma Allen Verco brought this to me when her and I had that conversation, and she's like, I'm all about bees these days and the microbiome. I'm like, what? I didn't even know this was a thing. So this is really cool to be able to talk to you. I know this is like your world. So thanks for making time for me, and welcome to the Two Nobodies podcast.
Thank you very, very much for having me, and I'm honored to be invited on the show. Yeah, I was just going to say, I'm a postdoc in Dr. Emma Allen Verco's lab, and so the lab overall focuses on the microbiome, and I do the bee microbiome component of that. So we're all microbes, we're all microbe obsessed, and I'm bee obsessed as well.
Yeah, for sure. Are bees? So just curious about the bigger problem. I think everyone knows that it seems like there's not as much of a bee population these days. Everyone's concerned about pollinators and such like that. But is it still a really, really big problem in terms of just colony collapse and just the number of bees and that sort of thing? Does that problem still exist? Or are the efforts that people are making, is it helping?
The efforts people are making are definitely helping, but there's still definitely a really big problem. The term colony collapse disorder is it's a little bit outdated now, and the term represents this phenomena that was happening in around 2007 to 2010, and where essentially colonies were just collapsing, colony collapse disorder, and they didn't know why. And it's that exact disease or disorder isn't still happening, but they're still experiencing really high mortality. And beyond bees, the bigger problem is that there's just a really huge decline in overall insect populations overall. Study done a few years ago in Germany showed 75% decline in total flying insect biomass over 27 years. So it's I'm sure if you talk to older generations, even when you're driving on the highway, you're seeing less bugs maybe hit your window, for example. There's just less insects out there. And so honeybees are a big focus because of their importance in pollination, but really a lot of insects are declining, and so it's important to kind of look at it. And there've been some theories in recent years calling it the insect apocalypse, and essentially the disappearance of all these insect species that are really important to overall total ecosystem function. And the bees, they play a really important role in pollination, and we use honeybees to support our agricultural processes. Honeybees, for example, support up to 1/3 of our global food supply in pollinating the crops. Without the honeybees, the crops wouldn't get pollinated, and they wouldn't be able to make food for us. So that's that's a really core reason of why honeybees are important. Other types of bees, native pollinators in different parts of the world, are really important for biodiversity and for supporting the health of plant species. And it's all connected.
Yeah. What's causing that big decline in the insect population? Like, what are those drivers from your understanding?
Yeah, I mean, there's huge debate on that, right? But for honeybees, or for bees in general, the big three drivers are pests and pathogens, pesticides, and nutritional deficiencies linked to loss in forage or biodiversity forage. So essentially, the bees are foraging on monocrops. Our agricultural system is set up for monocrops, right? So they're eating essentially the same thing day in and day out. You can imagine for human health, if you only ate fries all day, you wouldn't have the greatest health.
Yeah. So, okay, let's break those down. So, first one you said was pests and pathogens. So, what does that really entail?
Pests and pathogens. It entails macroscopic pests, things like ectoparasites. The big one for bees is something called Varroa. The actual species name of it is Varroa destructor, which is quite
Okay.
depicting. And they're these little tiny mites that are about I'd say 1/50th of the size of a bee, and they literally just like how a tick bites into the skin of humans, they just bite into the skin of honeybees and essentially eat them alive. And they can be really damaging to the populations. Bees can see them and they can physically remove them. So there's been efforts from a breeding perspective to breed hygienic bees that will basically remove these mites. So that's one approach for that. And then that's the pest side of that. And then the
So,
pathogen.
just a quick question on that, Brendan. In terms of those mites, do we see those more in those kind of monocrop like those situations, or are they just prevalent even in really diverse kind of landscapes as well?
They're prevalent all over the place. They're associated with monocrops for several reasons, and one of them is likely due to the exposure of pesticides and other agrochemicals that they're being co-exposed to in those monocrop situations. And essentially that lower, if you can imagine, if they're being poisoned essentially by a chemical, they're a little bit lazier, they're not grooming each other as much, they're not getting the mites off. So it all connects, and kind of when you see an increase in one issue, you often see an increase in multiple issues because many things are happening at once. And then so kind of
Okay, that makes a lot of sense.
connecting to that is the pathogen side of that. So bad bugs, bad microbes. It's the same thing with human health. If you have an infectious disease, you're not feeling great, right? You're not very
Yeah.
energetic. You're not gonna be addressing hygiene. So there's
Yeah, for
several
sure. Yeah.
pathogens that are really problematic, that have been characterized in honeybees, and one of them is American foul brood, and it's caused by a gram-positive bacterial pathogen. Its name is Latin name is Penibacillus larvae, and I studied this
Okay.
pathogen quite a bit in my PhD, and how it impacts honeybee health, and also why or when and how it
causes disease. It's a really fascinating microbe because it's found in honeybees, or it's found in the hive very commonly, but it doesn't always cause disease. So it's almost always there, doesn't cause disease. And then all of a sudden there's outbreaks of disease. So why does that happen?
Mm-hmm.
And the focus of kind of, my all of my PhD work and research was looking at how the microbiome could play a role in basically the outbreak of this disease. And it's, this pathogen has been studied for over 100 years, and we still don't know why outbreaks occur. It's just, it happens. And beekeepers and in agriculture, it's just, they respond by using antibiotics. And they solve the issue in the short term, but not the long term. And I'm sure we'll get to talk about antibiotics in a little bit. But yeah, it's really fascinating, and kind of my focus was on the microbiome and the role of the microbiome in transitioning between an asymptomatic disease state and a symptomatic disease state. And then another two pathogens that are really important are European fallow brood, caused by Melissococcus plutoneus, and a fungal pathogen, which nosemosis, which is caused by Nosema apis or serranae. And they're just different Latin names, but they're those are the main bacterial and fungal pathogens. And then beyond that, there's a ton of opportunistic pathogens. So just like how we can get food poisoning, bees can get a synonymous experience through opportunistic pathogens that they are exposed to, for example, on flowers or just when they're foraging.
So Brendan, it sounds like there are so many things that bees can be exposed to that can totally be deadly or affect their health. Hey.
Yeah, 100%. There's many stressors in their day-to-day life, including pests and pathogens, as well as pesticide exposure and nutritional deficits due to poor forage habitat.
Yeah, yeah. Now, okay, you started talking a little bit about the microbiome. Like this just sounds like a new field in general, but when I was doing a bit of research, it does sound like there have been people who've been researching the bee microbiome. But why is this like not making sort of the mainstream news, I guess?
Yeah, it's interesting. I also wonder why more microbiome research doesn't make the news as much. I mean, it is, it's being publicized more, and I think a lot of people are aware of the importance of the human microbiome. But I think
Yeah.
still our mindset of the microbiome and insects and animal health is not really at the forefront of our thoughts. Um, it's the honeybee microbiome has been studied since probably around 2010 when next generation
Mm.
sequencing came out and essentially made it affordable and accessible for researchers to look at most of the microbes. But the major thing with those technologies is that they can identify what's there based on essentially a fingerprint or a barcode, DNA barcode. So
Mm-hmm.
it gives you a sequence of nucleotides that they all match a different species, for example. But we don't have the databases to match those sequences or those fingerprints, let's say, to a known
Okay.
species because they haven't all been characterized. And today still there's a lot of biodiversity at the microbial level that hasn't been cultured or characterized in the lab, and we really don't know what they're doing. And not that microbiome studies have been, not that they're wrong, but their technologies were just lagging early in, from 2010 to 2015 period. And
Yeah.
yeah, so classifying those species were hard. So a big part of my research right now is actually using next generation culturomic techniques. So using our genome data to predict what the microbes need in terms of their nutrition or the media we grow them on, and then essentially dissecting out the gut microbiota, bringing those microbes from the honeybees into the lab, characterizing them, and then we can look back at the microbiome data and understand maybe their functional role in supporting honeybee health. a link I always like to make to studying the microbiome too is, we talked about the decline in insects, major decline in insects, right? They're macroscopic.
Yeah.
We can see them. So we know they're declining. But the same thing is happening on a microbial scale. We're just not seeing it, right? There's, the introduction of antibiotics over 50 years ago, the introduction of all these agrochemicals. Antibiotics, we know they have, antimicrobial capacities. Pesticides and other agrochemicals, they're not even tested for their antimicrobial capacities. So, they're only tested on the macroscopic life. And so, a big part of my research is essentially trying to capture that microbial diversity and study it and characterize it before it disappears. Because once it's gone, you'll never know if it was ever there.
So do we know for a fact then that these kind of antibiotics that are being first of all, how do they administer antibiotics to bees?
Yeah, well, we'll get into like how they, how you administer microbes in general to bees too.
Yeah.
But antibiotics, they just simply dust them in the frames. So
Okay.
beekeepers, they can essentially, take off the lid of the honeybee hive and dust the powder in, and it gets in all of the different components of the hive because of the way the bees move. They just, it gets on the bees, it gets in their food, it moves all around the hive.
Yeah.
And there's issues with residues left over for honey and things like that. So the beekeepers have to be very careful when they use it. But that's the essentially leading method to treat pathogens.
For you, was it bees first or was the microbiome first in terms of your own interest in research?
Yeah, definitely a microbiome. When I first got into research, I was looking at how probiotics in humans could reduce heavy metal absorption in the diet. For example, can you take a probiotic with a fish meal to reduce mercury absorption? That transition to looking at agrochemicals, how probiotics in humans could maybe prevent the toxicity of certain pesticides. So maybe you could eat non-organic food and mitigate the off-target side effects of pesticides without having to buy their organic. I mean, you're not addressing the core problem there, but you're helping people live with the issue at the time, right?
Yeah.
So all of that research really pointed me in the direction of honeybees because of how relevant pesticide exposure is to honeybees. They can be right in the field when the pesticides are being sprayed, right? So they're being directly affected by it. And we were wondering if we can use microbes in a similar fashion how we use probiotics in humans to improve bee health. So that was one of my goals, was kind of developing, the first honeybee probiotic with strain level defined properties for improving health.
That's really cool. I would just want to ask about the probiotics on the human side, because Emma and I had a really focused conversation on probiotics, and she's really enlightened me on the fact that really the gut for humans gets colonized between the ages of 0 to 3, like really early on in life. And she's like, the thought that a probiotic can have any sort of meaningful impact on colonization of the gut thereafter, she's like, there's really no strong evidence to suggest that it would. She's like, there might be some probiotics out there that have shown some effectiveness, but really, eat a spoonful of yogurt, you're probably not going to get some sort of meaningful colonization. You study probiotics as well. I mean, is there any sort of, do you want to build on that at all, or would you generally agree with that? Or
Yeah, I would agree that probiotics don't always colonize, but they don't need to colonize to have a benefit. Is what I would say.
okay,
Yeah,
that's interesting. Yeah.
yeah. So I've been a part of the International Scientific Association for Probiotics and Prebiotics. It's a annual conference that happens every year, and they have a student fellows component that I help run every year. And so it's really interesting because the general public's perception of probiotics is that that you take them and they help replenish your good bacteria, but that's not that's not how they always function. Um, they can help by a lot of ways. Even though they don't colonize, they might help get rid of pathogenic bacteria that are colonizing, right? Because of their bacteriocin production and antimicrobial peptide production that the good microbes are making. So essentially, there's a constant battle between good microbes and bad microbes in your gut,
Yeah.
and adding probiotics or good microbes can help get rid of or deplete those bad microbes. They may not necessarily impact your good microbes, and they may not stick around themselves, but they get rid of the bad microbes. And they can also help, for example, through the things that these good microbes are making, or these probiotic microbes are making, they can help restore your gut lining. So I'm sure in the public eye, leaky gut's a big thing, right?
Yeah. Yeah.
Leaky gut is simply intestinal permeability. So your cells should be, tightly linked like this. Nothing should be getting through, and
Right.
many stressors in life can cause kind of gaps to get through, and the probiotics can help tighten that back up. So even though they don't colonize, they can help your gut integrity and improve health in that way. Yeah. So essentially, I feel like the main message I would end there with is that probiotics don't need to colonize to have a benefit. Some probiotics can colonize and exert a benefit, but they all work a little differently, and not one probiotic fits, all situations. And also, a probiotic that is good for one person, or that has a benefit in one person, may not benefit another person. You got host genetics, and then you have, microbiome specifics. People have different microbiome compositions.
Yeah.
So all of these contextual dependencies on whether or not a probiotic may be beneficial.
Okay, Brendan, you're right now just like blowing my mind because I walked away from, and I think that's a really good distinction. Like I walked away from that conversation with Emma, like, okay, if it's not going to colonize your gut, and she didn't say this, so this is really my assumption and interpretation, but that if it's not going to colonize the gut, really there's probably no benefit. But like again, you hear so many people saying like, I took that probiotic and made me feel good. And Emma did acknowledge like can be some temporary benefits, but we really didn't get into like how that would actually, like, what those what those benefits would actually be. So what I'm hearing from you is that um that probiotics in some ways, potentially could help with leaky gut, with helping some of that binding that needs to happen with that permeability that's there in the gut walls. You also talked a little bit about your research with mercury. So like, is there a potential that probiotics can sort of help almost like detoxify or remove some of these kind of chemicals or whatever it might be? Or
Yeah. So the studies to date, I don't believe that the probiotics could pull the mercury out of you if you have, or the heavy metals out of you, like a chelation agent would. But they could
Mhm.
help in a prophylactic manner by preventing uptake in the first place. So
Mm.
um, yeah, the studies that have been done in like Tanzania, and they've been smaller pilot studies looking at how consumption of, for example, probiotics in yogurt could reduce mercury absorption from the fish there that are really high in mercury. Um, well, fish everywhere are high in mercury, but in certain parts of Africa, they're particularly high, and there's also pesticide leakage into the waterways and things like that. So
So,
yeah.
okay, so people are going to listen to this and they're going to be like, they're either going to think maybe, okay, what probiotics should I even focus on? Or now should I like, what's your sort of takeaway for folks then, like in terms of how they incorporate probiotics? Is it more through like, what about fermented foods? Like, or is it actually taking a probiotic supplement? Or like, I don't know, how do you help someone navigate this question? Because I'm sure people ask you about this.
Yeah, for sure. Um, the first thing I always try to say is that yogurt isn't probiotic, and kombucha is not probiotic, kefir is not probiotic. None of these things are actually probiotic. Um, People often will call those fermented foods probiotics, and they're not. They're fermented foods, and the distinction is should be clear. And I think we should make it more common in you know public eye and public language discussing it, because fermented foods can have a benefit. We've been fermenting foods for thousands of years. We know that humans do that. It's been a great way to
Hmm.
preserve a food, and there's also health benefits associated with it. But for a microbe to be a probiotic, it has to be distinctively characterized at the strain level. and it has to have, double-blind, placebo-controlled clinical studies done on it. It has to have multiple, right? And it has to show a distinct benefit for treating a distinct disease or a distinct disorder. It's, probiotics aren't this like one size fits all magic, potion. And I feel like that's when you start thinking of it that way, or you start communicating it that way, it becomes less clear of how they are really beneficial. So, for just a really simple way to remember it is, if you look at the label and you see something say, let's say Lactobacillus acidophilus, that's the genus and species name. After it, you should see a strain name. That strain name shouldn't be italicized. It should just be like, sometimes it's alphanumerical code A123. That's the most important part to look at when considering probiotics, is that strain name. So there's the most well-studied strain or probiotic strain is Lactobacillus bacillus rhamnosus, GG. And you'll also see it shortened as just LGG. And that study that probiotic has been studied the most out of all probiotics, and it has tons of clinical evidence for supporting essentially gut health and IBS symptoms, and can be immunostimulatory in certain circumstances and have certain health benefits. So if you remember the mouthful of name I just said, Lactobacillus bacillus rhamnosus, GG, and then you have another strain, maybe it's AA. GG and AA do not necessarily deliver the same benefits. So often fermented foods can have a variety of strains, and depending on where you make those fermented foods depending on the geographic location, there can be different strains in that area, right? And they're not all going to have the same benefit. So you can't expect them to reproducibly deliver a health benefit when they're not the same strain. And the same thing in the honeybee world is that, or you're really in the animal world in general, probiotics for livestock, they haven't been as regulated as the human side of things. You go into the supermarket now, and our regulations are pretty good, if you in Canada at least, and in many places in Europe. If you go into the yogurt section and you see something say probiotic, you can almost always guarantee that it will say the strain name that has been studied in a clinical study to support whatever it's saying it's a probiotic for. Advertising as a probiotic is and you're not defining at the strain level, isn't really allowed or feasible in the human side of things anymore. But in animal probiotics, it's still almost all of the products that are available still aren't specifying the strain name of the probiotics. So they may not, first you don't know if they've ever actually had a study done on them. You don't know of the real scientific evidence, because they're not defined at the strain level. And so you don't know if they actually have scientific evidence, and it's not reproducible. The thing we've been trying to do with developing honeybee probiotic is characterizing them at the strain level. We've been working on a mix of 3 probiotic strains, so a triple strain probiotic, strain-defined benefits for treating infectious disease in honeybees, and through that way improving honeybee health.
Okay, now on the bee level, though, is I guess the idea with the probiotics is not necessarily to colonize the gut in this situation. Again, it would be a different purpose.
Yeah, exactly. So the 3 strains we use are from different sources. So one is from a plant. It's Lactobacillus plantarum, LP39 is the strain, and it is originally derived from a plant. And honeybees' association with plant and their co-evolution with plant kind of made sense in that way. Another strain was isolated from a healthy honeybee, and it was actually part of their gut microbiome. So it was a honeybee derived so we've had a lot of name changes in the microbiology world. Its name now is Lactobacillus kuhnkeii, BR1. And that was derived from the honeybee. And then finally we have a the third probiotic strain was Lacticaceae bacillus rhamnosus, GR1. And it's actually a human characterized probiotic that is used to support vaginal health in women. And the idea is, honeybees are a matriarchal society, and they're a full hive of females essentially, and it kind of makes sense in that way. But beyond just the intuitive sense of or selection of those strains, we've shown they actually have functional properties in bees and also in other insect models. And the way they work is through stimulating the immune system, the innate immune system of honeybees. So it essentially boosts their immune system, so it enables them to fight off the pathogens better themselves using their evolutionary arsenal of different immune factors. So its benefit extends beyond just looking at one pathogen too, and we still need to study this more, obviously. But the concept is that if you can support their immune system, it will support them fighting off many different types of pathogens.
Got it. Yeah. Would that similar principle translate to humans then as well? Like when we talked about probiotics in terms of just stimulating the immune system, like does that same effect, would that same effect happen for humans?
Yeah, and I mean that's been a large component of many probiotic studies, is characterizing the immunostimulatory benefit of select probiotic strains. So, they don't colonize, but they do get into your gut. They stimulate your intestinal epithelial cells to upregulate production of an important immune factor that helps control, let's say bad bugs.
Yeah.
They can work in that way, and there's lots of studies supporting lactic acid bacteria and bifidobacteria in supporting a healthy immune system in the gut, and that has systemic effects too, right? So to date, if you're focused on human probiotics, to date, the large majority of human probiotics have been lactic acid bacteria. So things like lactobacillus species and bifidobacteria, right? And now you're seeing this whole new generation, and I'm sure you discussed some of this with Emma as well, but you're seeing this whole new generation, kind of next generation probiotics, right? And one of my favorite next generation probiotics is Akkermansia muciniphila.
Yeah, she talked about that one. Yeah.
So I mean, that one's and that one does colonize. That one, it does colonize, right? Because it's derived from a human. It's originally derived, like if you extract or isolate a beneficial microbe from one human, culture it, and then supplement it to another, depending on their context, depending on their genetics and their microbiome background, it may or may not colonize, but it has a better chance of colonizing than, let's say, a bacteria that was isolated from the soil, because it's already adapted to living in the gut of a human. And there's several next generation probiotics that I think are going to be coming out and becoming much bigger in the next decade or so. Akkermansia is one of them. And also functional fecal microbiota transplants. I'm sure you talked about that as well.
Yeah.
Transplanting an entire community instead of just looking at like one microbe. It's like, how can we take a good microbiome and transfer that good microbiome to someone who doesn't have a good microbiome to improve their health outcomes?
Yeah. Okay, so let me just, I just want to reflect on something. So if the goal is not really about colonization with some of these probiotics, then I think like everything you're saying makes a whole bunch of sense. If there is a goal of colonization, from what I understood from Emma, is that it's not as simple as just plugging and playing, right? Like just because you're low on one microbe doesn't mean you can just put that microbe into your gut and then everything's going to be good. Like diversity is really important, but it's also like the ratios of these different microbes, like it's a community and every community is very different. And it's like she's like that hasn't been really figured out. So like even like fecal biotransplants, like have shown to be beneficial, but in terms of colonization, she's like it's not a direct sort of plug and play sort of thing. Is that fair to say? Or
yeah, I would agree with that fully. It's not plug and play, and we also don't even understand how it's working sometimes. There's been cases where they actually take a fecal microbiota the feces from one human, and they will they will put it through a filter.
Yeah.
So it gets all of the metabolites or things that the bacteria and microbes are making, but none of the actual microbial cells themselves. And then they transplant just that extract into a diseased participant in a clinical study, and it has a health benefit. You're not putting any of the microbes, you're just putting their metabolites, and it somehow resets the other person's microbiome into becoming healthy. So there's things we don't understand. There's a lot of hypotheses on that. That could be due to bacteriophages that basically you can't filter them out because they're so small, but they may play a really important role in regulating the microbes that are there. Bacteriophages, they can have lytic effects or bactericidal effects on certain populations of bacteria in the gut, and they just reach an equilibrium where they cycle through their populations and keep everything in balance, and sometimes that might be the important factor.
Okay, let's step back one more time as well. The big problem that you're really trying to solve, how would you summarize that for people?
Trying to address microbial imbalances in bees and to improve greater ecosystem health, and also to support sustainable agriculture. That's the real focus of my research. And the role of microbes, the role that microbes play in supporting bee health is relevant and similar to the ways that microbes support human health and the health of many other animals as well.
Okay, and so how do you even break down that problem? Like when you're thinking about like what are the different components of that problem that you feel like you need to address, I guess?
Well, so when we first started looking at this, we looked at it like this. Bees are being affected by three things, right? We discussed pathogens, pesticides, and let's say habitat loss, just to summarize that third one. We can't really address habitat loss single-handedly, but we can address the other two. We can address pathogens and we can address pesticide exposure, or we can try to address that through developing a probiotic to improve immunity, and a probiotic to detoxify pesticides from honeybees. And that's been the focus of our work, is essentially trying to address 2 out of the 3 main causal factors of bee decline. So we've had really good success with doing that, in developing one probiotic I mentioned earlier, the LX3 triple strain probiotic for honeybees. And that's shown to be immunostimulatory to the bees and help improve their colony buildup. So they basically, it reduces disease and increases the population, the number of bees in a hive.
Okay. How do you even study, like, how do you gather their microbes? Is it, I guess, through their fecal matter, or like what is it like? How are you gathering this?
From bees, how you get the microbes is generally dissecting their gut. If you're trying to get an entire population, you do have to have some sacrifice in some of the bees, and you just basically have to sample a number that is representative of the hive, and that's anywhere from 3 bees to 30 bees per hive generally. And there can be anywhere between 10,000 to 50,000 bees in a hive. So you're not really impacting the overall population, but for doing science, sometimes there needs to be sacrifice.
Yeah. Okay, so you got to actually dissect them, and then what do you do from there? What's the process like then?
Process after that is basically homogenizing them and then plating them out, or making cultures of them in the lab, and basically to isolate colonies. Colonies of bacteria or colonies of yeast, and then from there you have to make sure the culture is pure, and then you make a freezer stock, and then once you have a freezer stock, you can just keep going back to that freezer stock and studying it as much as you want without having to disrupt the colony or anything like that. Another way, so that what I just mentioned is the culture based method, right? The other way to study everything going on in the bee, because you're not going to be able to culture everything that's there. We can try, and we do try to culture everything that's there, but there's always things that won't grow. So another way to study that is by doing microbiota sequencing, things like Illumina, MySeq, next-gen sequencing, and basically that allows you to look at the barcode or the fingerprint of those bacteria and of those of the entire community, so you can see everything that's there. And the issue though is that we don't always know what those line up to. We can tell that they may be related to a species that has been characterized, but we don't know their actual species identity. And so I made a database a few years ago to try to address this actually. And it's kind of so we call microbes that haven't been characterized, we call it microbial dark matter.
Okay.
Unknown, right?
Okay.
It's like the dark matter of space, but the dark
Yeah,
matter
yeah.
of microbes. And so we know what their fingerprint looks like or their DNA barcode looks like, but we don't know, their function. But to enable tracking of those microbes or associating those unknown microbes with, positive or negative health outcomes, I assigned a placeholder ID to basically the barcode matching these unknown microbes, and there ended up being 600 and some number of them, 621 I think, unknown species that I essentially just gave a placeholder name so that other researchers, when they are studying it, they can assign that barcode to that name, and we can start associating these unknown microbes with different health outcomes of interest. And then ultimately, when someone has figured it out, or when they ultimately have been cultured in a lab, we can go back and be like, oh, okay, hey, this microbe's really cool because it was found, it was always present when the bees were experiencing this certain disease.
Yeah.
So you can go back and be like, okay, now we've isolated, and now we can interrogate its genome and see like, hey, what virulence factors or what things are in its genome, and how could we address this, and how can we improve microbial balance through keeping it at bay.
Yeah, that's really cool. I had David Good on a few not too long ago, and he's obviously studying the Yanomami population. I'm just curious about, like, from from the bee side of things, are there like ancient or older kind of bee populations that maybe have a unique sort of microbiome fingerprint at all, or
Yeah.
yeah, just curious if there's that same sort of analogous, like where you have sort of these indigenous populations and they have these incredible microbiomes, very diverse sort of thing. Is there similar Are there similar situations with, I guess, the bee population?
Yeah, and I'm very interested in that. I'm glad you actually brought that point up because,
Okay.
yeah, in my opinion, many of the bee populations that we have, that are in beekeeping operations, they've been exposed to a tremendous amount of antibiotics and
Yeah.
pesticides, transgenerational exposure, right? And that's really important because there's some microbes that are vertically transmitted from the queen to the offspring, and then subsequently to the next population. Um, by having transgenerational exposure to, let's say, a given antibiotic, it can sometimes end up curing or reducing vertical transmission of really important symbionts. And one of them is an intracellular symbiont known as Wolbachia, if you've ever heard of that. It's
No.
studied a lot in mosquitoes and also fruit fly research, or Drosophila melanogaster research. And so just to give you kind of a glimpse of how important this microbe is, it's not really considered part of the microbiome. It's because it's intracellular. It's not in the
Mm,
gut. It's just in their cells, and
mm,
usually it's in their actual gut cells the most. So it's in different cells at different concentrations. So Drosophila, with this intracellular symbiont, which almost acts as like a secondary mitochondria in a way,
okay.
it gives it tremendous resistance to pesticides, tremendous resistance to infectious disease. And I studied fruit flies basically that were Wolbachia free versus Wolbachia colonized,
Yeah.
and the Wolbachia colonized ones were just like, it was amazing at how much more robust they were. And so the thought is that transgenerational exposure to these antibiotics in honeybees could actually have cured or got rid of this important intracellular symbiont. So in fruit flies, the way that you get Wolbachia free Drosophila is through 3 generations of exposure to tetracycline.
Okay.
The most widely used antibiotic in beekeeping is oxytetracycline, which is just a tetracycline derivative. So the exact same antibiotic that you use to cure this really important symbiont is in fruit flies is used in the beekeeping world all the time. So it is my thought that the Wolbachia probably plays a really important role. And that's I'm spitballing, just focusing on one single symbiont, but this could have happened to other ones, right? So I'm really interested in sampling bees in kind of hidden spots or hidden pockets in the world that haven't been exposed to the industrialization in general. And not only looking at honeybees, but also looking at other other related bees, other bees in the corbiculate family, or that's just a fancy way of saying pollen pants. They collect pollen. They have little pollen pants on the side of their legs. So we have some collaborations of basically trying to collect bees from different parts of the world: Peru, Nigeria, and actually Cuba. And so we're in the process of actually extracting their microbes and looking at maybe how some of those microbes from these more ancient or less industrialized microbes, is a better way of putting it, could help improve the health of our industrialized microbes in a similar fashion to what David Good is working on with the Yanomami, right? And it's just understanding kind of how industrialization has really messed up our microbiomes and also the microbiomes of animal life around us.
Yeah. How vulnerable is the bee microbiome to just other environmental factors? Like, forget about the 2 main or the 3 things that you really talked about, but just other sort of, pollutants in the air or whatever it might be, other environmental sort of exposures.
Yeah. We don't know.
Okay.
We simply don't know what the grand or totality of extent is in terms of environmental exposure to different chemicals, the impact on the microbes in the bee, and how they may be leading to a microbial kind of apocalypse situation right under our nose, but you don't see it because it's not macroscopic. And is a good segue into this is that's what we're trying to do with the Canadian Bee Gut Project. We're trying to, and I'm wearing the shirt to publicize today.
Oh yeah, love it. Yeah.
So we're essentially this project is a crowdsourcing based project or initiative aimed at collecting bees all across Canada to determine a baseline for their microbiome. So to just see what microbes are in bees in different areas, and to look at them now before it's kind of too late, before things start disappearing completely.
Right.
And who knows, maybe that has already happened to an extent, right? But we're developing a baseline to essentially be able to compare to, because we can look at that, we're gonna do this within the next 3 years, and then in the next 20 years we can look back and say, hey, we determined that the microbiome profiles in 2025 looked like this. 2045, they're all of a sudden they've lost even more microbes. Hopefully it doesn't get to that. But the point is to basically profile and catalog these microbes well we can, and develop kind of more microbial ethos and thought in developing sustainable disease management practices with the microbiome in mind. And probiotics, in my opinion, is one route to that. Ultimately, we need to start thinking about how bees are impacting other parts of the system, of the ecosystem, and other parts of agriculture and things like that, and also vice versa. Bees, in my opinion, are such a great model to study in terms of microbial transition, because they go out and they forage and they touch so many different things in nature. And so if you think of bees and different pollinators, you can refer to them as the pollinator assemblage. So bumblebees, carpenter bees, all sorts of different bees, they're all landing on these same flower pads. They're all sharing microbes, right? Bad microbes, good microbes.
Yeah.
If you can help balance the microbiome or improve the good microbes in honeybees, you can potentially influence all of these other native pollinators and other parts of the pollinator assemblage by simply just you're giving good microbes to bees. They're getting on these flower plants, they're sharing good microbes with the rest of the pollinators, improving overall health of all pollinators, and hopefully helping to address the decline in overall insect populations.
Does that assume though, like in that case, that the probiotics are giving those honeybees that they will be beneficial for those other pollinators though?
Yeah, that's that. We haven't reached that point for that.
Yeah.
That's
Yeah.
it's a perfect reason or perfect point that we need to look into more. But there are studies showing that honeybees and bumblebees and other pollen collecting bees do have a shared kind of core gut microbiome. So they
Okay.
have many microbes that are beneficial to them, not in a complete harmonized way, but a certain subset have beneficial functions in many different bees.
Hmm.
So I think there is potential to do that, and it kind of makes sense from an evolutionary standpoint. They actually have been co-evolving because they land on these flower pads,
Yeah.
right? And they've been co-evolving their own microbes with each other, and also the host genetic factor there too. The same thing could be said with plants. Plants and pollinators have co-developed. They've probably co-developed to prefer certain types of microbes. Certain types of
Yeah.
microbes benefit plant health. A good example of this that we're in the process of studying is I mentioned earlier, one of the strains in the probiotic we're working on for bees was derived from a plant.
Mm-hmm.
We're seeing if that probiotic can have plant stimulating properties as well. So if you give it to bees, you improve their health. Can you also improve the health of, let's say, a crop?
Oh, yeah,
Can
that's
you
so cool.
increase yield, right? What if
Yeah.
this thing had a plant growth stimulating properties?
Mm-hmm.
So trying to advance things using microbes in a thoughtful manner, and without adding more chemicals to the environment is the overall goal.
I got so many questions. I don't even know which way to go right
uh,
now. Because I want to
yeah.
I want to sort of focus on the Canadian Bee Gut project, but also just something that's coming to mind is I had somebody who we talked to about plant to plant communication, and how plants have this amazing ability to communicate. What I never got into though was just, is there an interaction between plants and insects? And I wonder, like, could there? I don't know. I don't know if you know where I'm going with this, but like, could, as you said, in one way, if you're giving probiotics to the bees, could that have some sort of mutual benefit to the plant? But could the flip also be true, in that, are the plants also benefiting the bees in it, beyond the pollen and providing that kind of direct benefit to the bees, but in another way, I guess?
Yeah, I think it goes both ways. Just like I mentioned earlier, like if you start looking at things from an ecological standpoint or from an
Yeah.
evolutionary standpoint, you can make sense of a lot of things instead of
Yeah.
just blindly being like, this we want to give this microbe because it's good. It's like, why is it good? Like what is the rationale for that? If you start looking at things like, oh well, this microbe has co-evolved alongside honeybees, alongside bumblebees, alongside these other pollinators, and alongside the plant, it's like, starts to make sense. And it also starts to make sense that the plant would want to maintain the health of pollinators, because those pollinators are, they're helping
Absolutely. Yeah.
plants, asexual or sexual reproduction in plants. So the microbiome of plants, in my opinion, does play a big role in honeybee health through impacting the honeybee microbiome and also the microbiome in their diet, because the pollen and nectar, they have microbes in them, right? Bees essentially eat in a completely fermented diet in a way. It's like there's
Mm-hmm.
microbes in everything.
Yeah.
um so yeah, I think too also, and we need more research though to like really understand the mechanisms of all this, right? Right now it's
Yeah.
just, we have association studies
Yeah.
associating the microbiome of certain plants with poor health in honeybees, but we don't know exactly what's going on. Like, why is that? Is it the plant is less healthy, so it's delivering less nutrients to the bees?
Mm.
That's another way of looking at it.
Mm-hmm.
Or is it simply those bad microbes are at higher abundance on the plant and their bees are being exposed to that? another reason for pesticides, another kind of deleterious effect is that they can impact plant microbes as well, right? And the major thing with this is that it's not being looked at. I wrote a review article like last year, and it's over. I think it was estimated over 90,000 agrochemicals are actively registered. It was in the U.S. alone, and almost none of them are registered as an antimicrobial agent. They're registered as a pesticide, which have different regulations to what testing they have to be done, where what testing has to be done on them before they can, be sold. And a lot of them, for where honeybees are at risk, it's like they have to show that they're not those chemicals aren't toxic to honeybee cells or bee cells, right? But not bee microbes. You're not looking at the whole. It's that in that way. It's not evaluating the entire picture, right? And it comes into things like the holobiont theory or holobiont evolution, and looking at all components of an animal host, the host genes, the microbiome genes, and how they work together as a single unit.
Yeah. Are you through the bee gut project? Are you going to be able to identify bees that have maybe come from places where maybe there's more organic farming or around, or because I would imagine that maybe the bee microbiome could be influenced by like the soil health around plants or things like that. So are you going to be able to decipher like where these bees are coming from and like what they've been exposed to, I guess, to some degree?
Yeah, so that's a really fun or exciting potential of this project, is that we by through doing it with crowdsourcing or getting beekeepers to help us, right? They know their local environment, they know what the bees have been exposed to, and they know what crops are around them, and they can answer all these things in the questionnaires that we give them, and we have that metadata now that we can associate with the microbiomes when we profile them. So if, for example, the bees are surrounded by an organic um, farming practices.
Yeah.
They're all organic around that hive. And it's like we see certain beneficial microbes are super high in those bees, and overall the bees had low mortality. It's like, hey, there seems to be an association here, right? And then you can start looking at that more. And I think more importantly is kind of the identification of certain crops or certain pesticide usages that may be deleterious to certain bee microbes. It's like, hey, when they're beside these crops, which we know are, they're sprayed with this sort of chemical, it's like all of a sudden those bees they've completely lost this one beneficial symbiont, and also they had really high mortality. Those things could be linked, and it helps direct us to really seeing what the core problem is, and helping develop a solution to it.
Any associations you've been able to make so far with the Bee Gut project, or not yet?
We actually started the project just a year and a half ago. So we've
Okay.
collected over 800 samples now at 91 locations across Canada. So large number of samples, and we're in the process of simply dissecting the microbiota and doing DNA extraction and sequencing on it. And we hope to have answers by 2025.
All right, I'm gonna have you back in next year then for sure,
Yeah.
to see what kind of conclusions you made. Curious about just like the similar characteristics between like the human microbiome and the bee microbiome. I
Yeah.
know with the human microbiome diversity, microbial diversity is really important. It sounds like, or there's been associations with some positive health indicators with microbiome diversity. Are there any similar characteristics between the two microbiomes?
Yes. Yes and no, right? I mean, obviously humans and bees are much different,
Yeah.
but the way the microbiomes work are very similar. And it's,
Okay.
humans or bees have been proposed by various researchers as a good model for studying humans, especially because bees are social and they develop social interactions, and so they share microbes in that way, right? And it's how humans develop social interactions, share microbes. Um, Microbes they differ, but they show the same patterns. So one of the patterns is that bees have core gut microbiota. They have a core community of microbes that are found in almost all honeybees. Just like how humans have kind of a core microbiome that is found in most individuals, especially regionally, grouped individuals.
Mm-hmm.
And so this is unique, and it's something that's not actually apparent across all of animal life, especially not all of insect life. Many insects actually don't have a developed core gut microbiome.
Okay.
It's that they just essentially take the microbes that are in their environment. They rely on those as just simply like passing through, and they serve a function to their digestive system and overall health just as they pass, and they respond to the environment in that way. Honeybees have a microbiome composition that it's passed vertically and horizontally to maintain basically that same composition. And you can look at a honeybee, in Europe and here in North America, and they have almost, considering a healthy honeybee, they have almost the same microbiome composition. And just like humans too, going down their digestive tract, they have the microbes start at lower concentrations, 10 to the 3, or maybe 1,000 cells, and then when you get all the way down to their rectum, which is the equivalent to our colon, numbers are all of a sudden up to 10 to the 9, or a billion cells per gram of fecal matter, for example. Obviously, it's all at scale; it's much smaller. But the concentrations of microbes are all very similar. Yeah. And so the main, another really important thing to think about of these core microbiomes, or why core microbiomes are important, especially from an evolutionary standpoint, of social species, or of social animals, is their ability to allow the animal, or the host, to interact and thrive in a variety of environmental conditions. Honeybees, for example, are generalists. They're actually evolved to forage on a wide variety of plants, or a wide variety of flowering plants. And that's not true for all bee species. Through that, they're exposed to a number of natural plant toxins. There can be, humans know not to eat certain things, certain berries in the wild. They can make us sick, right? The same theory can be applied to bees. And they actually, their studies have shown that certain microbes, or certain symbionts in bees, help the bees break down these toxic plant chemicals.
Yeah.
So it's not only the environmental chemicals, right? If we're taking that completely out of the picture, there's just natural compounds, secondary metabolites in plants that are toxic to animal life, and basically the honeybee microbes help break down those toxins. And a stat from humans, and there's no equivalent stat in honeybees, but the same principle applies. Humans have something like 25,000 genes, unique genes, whereas the microbiome of humans, if you were to enumerate all of the unique number of genes encoding a protein of interest, it's like over 3 million. So far more number of functional proteins and enzymes that the microbiome can make in comparison to the human, right? And same thing with bees. They have much more genetic capacity in the microbiome, which allows them to adapt to different environmental situations and stimuli, and also resist or tolerate environmental stressors. And that's one of the most important kind of roles of the microbiome, in my opinion, is that increased genetic information. I mean, the basis of life is DNA, right?
Yeah.
So if you have more genes, you have more you can encode more things to metabolize toxins, for example, or maybe to make nutrients. A topic that we haven't discussed is nutrient production by the gut. One of the reasons honeybees are having, their populations are struggling so much, high mortality, is nutrient deficiencies, which have been attributed to monocrop cultures and poor diets. But another factor that could be involved with that is microbiome composition. If they have a dysbiotic microbiome, which is an ambiguous term, but let's just say not balanced microbiome, they may not be making all of the essential nutrients or essential vitamins in their guts that they normally would. And that can impact health as well by causing nutritional deficits.
I didn't know that. Okay, so maybe I didn't know this, that the microbes in our gut you said can make nutrients, can make these vitamins?
They can make lots of vitamins. Yeah.
Okay.
There's a actually it's a 2015 study and another 2019 study by the same group. It showed they used computational techniques and prediction algorithms basically to show that the microbiome of humans in a healthy human could make B vitamins, like certain B vitamins, up to 70% of your recommended daily intake could actually be supplied simply from your microbiome. So, and it wasn't all B vitamins, some of them were less, but certain nutrients could be made all at abundance in your own gut. So it kind of makes sense. You have a friend that only eats fast food and it's like they're in impeccable health. It's like how does this happen, right?
Yeah.
And it's like there's many anomalies that you see like that through the human experience, and it kind of makes sense to how that can occur.
Because they could be having these microbes that are providing them with the important nutrients, I guess.
Exactly, yeah. They subsist on nothing but pasta, and they just have excellent health. It's like, how could that possibly be?
But I imagine those those poor diets, they'll probably obviously have an impact on those beneficial microbes too, right?
100%, and it goes both ways. And another thing, your microbiome can make vitamins, but it can't make minerals. Minerals are the periodic elements of life or of Earth, right? You can't make them; they're constantly in transition.
Yeah. You talked about some of those that bees are really social, humans are really social, but just with like, does the microbiome have an effect on just social behavior at all of bees?
Yeah, the whole gut-brain axis in humans has
Yeah.
recently been studied. Yeah, and I saw that research coming out now is so fascinating to me, and it just makes so much sense too, because you think of yourself eating a food you love. It's like, do you love it or does your microbiome love it? And it's like creating these chemical signals to make you feel absolutely amazing after it. And how just certain people have depression and all sorts of other neurological conditions is like, how are they linked to the gut, and things that we have been overlooking for so many years. There's far more research in humans, and there's not as much research in bees or insects in general. But there is good evidence to suggest that they have anatomical features similar enough to human synapses and neurons connected between the gut and the brain, or what their equivalent to the brain is, and that is likely a factor in their behavior. We need to study it more. One thing that we've been getting into for the past year or two is actually taking bees into the lab, feeding them different microbes, and then measuring their behavioral responses. It's very difficult to study the social behavior of bees because bees' social behavior are so complex. It's just absolutely like, a lot of the time when we're doing these things, it's like we start to see just behaviors of bees in general have nothing, well, maybe nothing to do with the microbes, maybe something, but it's like you start just sort of like, wow, this is a cool behavior by itself. Like, we're not even testing control versus probiotic. It's just like, what are they doing? It's like, how, and also like, how are they communicating, right? It's so fascinating. I think going forward, what would be really cool to develop, and is what we're trying to develop, is a true germ-free model of honeybees. So basically, they do not have a microbiome, right? And you can do that, for example, in mice. Mice have been derived completely germ-free, and then you can put back in, microbe A, B, C, and measure their phenotypic or behavioral response, right?
Yeah.
We're trying to do that with bees, but not sure if it is possible. There is evidence from non-social bees and other essentially non-honeybees, non-honey producing bees. There's evidence that they actually can't develop without a microbiome. So you know how we were just mentioning nutrients. The microbiome is actually provisioning them vitamins that are or steroid synthesis that is actually essential to their development. and without it, they simply cannot. They just they cease to develop. So it may be that we have to overcome that. We might have to find which of these microbial, or like what vitamin or what compound is the microbiome making. As like, can we supply that? If we supply that in their diet, can they then be derived completely germ free?
Yeah.
We know mice pretty good. We know exactly what they need. So it's like we've been able to do that, but we're not quite there with bees yet.
So I imagine we probably don't really know the answer then around like, do bees behave differently when they're exposed to just like monocrops versus various sort of diverse environments? Do we have a sense of their social behavior with those kind of different situations?
That in there's been plenty of studies comparing like their health between those two different environments, but
Mm-hmm.
nothing comparing their actual behavior. Something I did want to mention that's kind of very on topic with this was one study I saw a little while ago, and it was comparing um, rewilding of the microbiome essentially. That topic has come up in human
Yeah.
health a lot, is
Yeah.
rewilding. And it's, if you take a colony of bees that have been exposed to monoculture for whatever 2 years, long enough for it to have an effect on their microbiome. You profile their microbiome, it looks not balanced. They have overgrowth of opportunistic pathogens and not many good microbes. You then take that colony, you put it into kind of a pristine environment that has no agriculture associated with it,
Mm-hmm.
just it's completely natural. And they actually have shown that the microbiomes can repair or recover to an extent. It's not the exact same as before exposure, but it does appear that they get a lot of beneficial microbes back, or that they somehow regain homeostasis. And it could be through what we were discussing earlier with the pollinator assemblages in the flower patches, right? It's like some of those shared core good microbes in other bees could have been shared on those flower pads or from
Yeah.
physical contact with one another. So
Okay.
I guess
Yeah.
that if you think of sociality and physical contact and touch and things like that, that could be one type of behavioral response, right? Um, and which ultimately affects exchange of microbes.
Yeah. Yeah. I was going to also ask you, Brendan, like folks are obviously getting their gardens ready. There's a lot more
Yeah.
awareness about having flowers for pollinators, all this kind of stuff. Just based on like your research and the things that you focus on, advice to like the gardeners, is there anything that folks can be doing to maybe support bees from a microbiome perspective or just their health overall?
Yeah. Flowering plants, angiosperms, a diverse mix of flowers if you can. I mean, honestly, different flowers often are visually appealing to us anyways, and
Mm,
I think it's kind of a natural thing across many species, right? Plant as diverse of a garden as you can. Essentially, the more diversity you have, the more diversity the bees will have in their diet, right? And that alone will help support a diverse microbiome that supports their health. That's the best kind of suggestion that I could give at current, because we're still learning so much, and it's
mm,
hard to say what to do besides simply trying to emulate what we know is evolutionarily best, right?
yeah.
Yeah. And if
Yeah.
yeah, I was gonna say, if you have like ponds, you have bird ponds for birds to come and feed water on. Sometimes bees like those ponds as well, and they have little sticks you can buy in the ground where it's like it's a little platform bees can land on, and they don't drown in the water, but they
Oh,
can
okay.
actually get a little bit of the water. Something that people don't think about is the fact that bees need water.
Mm,
And there many water sources are kind of difficult for bees to get in. So
yeah.
they rely on, when there's rain and things in small puddles, and they like that. But if you can get a little water reserve, that's helpful.
That's good to know because living here in Alberta, like we're definitely facing some huge drought risk situations. So just thinking about, it's still a little bit early right now for the bees to kind of come out, but they will in the next few weeks. But yeah, we haven't had a lot of snowpack here in Alberta. Like it's definitely a concern for many other things like wildfires and stuff. But just thinking about the
Yeah.
pollinators, yeah, never really thought about that.
Yeah, yeah. There's many things that I think we don't think about with honeybee or with bees in general because they're just so complex, right? And it's like you
Yeah.
start it's like right in front of you, but you just don't see it.
Yeah. Just want to ask you about honey.
Yeah.
Is there, obviously people see a lot of health benefits from honey. Um, there are no microbes in honey, are there? Like, is it sterile or?
So yeah, honey itself is, I wouldn't say sterile because there can be microbes in them, in honey. Usually they're just in their spore form. But the way that we process most honey, through pasteurization and things like that,
Mm,
and even just filtration processes, it gets rid of most of those microbes.
yeah.
And most of the microbes that would ever be found in honey aren't actually that dangerous to humans. Um, and they're inert. They're not growing because they can't grow in the honey, right?
Mm,
But the making of honey, microbes do play a role, and I think that's something that is overlooked a lot. And it's not studied enough. And there's only so much time in the day. There's so you get into the microbiome world, and especially honeybees. It's like there's
yeah.
just so many
You
things I
can't
want
do
to
it all,
study.
Brendan. We get it. You can't do it
No,
all.
I really, this is
Yeah.
something I've thought about for years though, and I've never had
Yeah.
a chance to study it myself. Is looking at how microbes or good microbes in honeybees can improve the medicinal qualities of honey. So
Mm,
everyone knows Manuka honey is
yeah.
really beneficial. The reason Manuka honey is so beneficial is from Manuka honey is made from basically bees foraging on tea tree plants, and
Mm,
it's a compound in the tea tree. I believe it's methyl glyoxylate, and it's actually metabolized by the microbes.
mm,
It's a microbial metabolite that makes the Manuka honey so antiseptic. It's not actually, so where we're going with this is like if you didn't have the microbes, you wouldn't have really true Manuka honey. The
right.
microbes are what makes Manuka honey so good, so and have such medicinal qualities. And I often wonder if we don't have tea tree here. It's primarily like in Australia and New Zealand, right? But other plants we have here that people will have long believed that have health benefits. It's like, is there some sort of microbial factor involved there,
Right.
right? Um, can the microbes that are starting in the honey or in the glands of the bees, it's when they're secreting the honey, um, when they're mixing everything and having that initial fermentation before the honey kind of sets and then
Yeah.
becomes honey. It's like, can the compounds made during that time um, be influenced, because, for example, by supplementing the hive with certain probiotics or certain beneficial microbes?
Yeah.
And same
Yeah.
thing goes like if they don't have those microbes, it's like, is the honey as beneficial? Are we creating through agriculture and the use of broad spectrum antibiotics and pesticides? Are we making honey less good from a health standpoint?
Yeah. How far are we, Brendan, from actually? Because I didn't get the sense that we're at that sort of commercial stage with probiotics for honeybees. Am I right about that?
Yeah, we're working on commercializing it, but it's just working on a way to get it into the hive and everything. So
Yeah.
we've done several studies now in Ontario here in Canada, as well as California in the States, looking at different delivery methods.
Mm,
So how you get good probiotics into honeybee hives is very crucially important, and it's been done wrong in many studies and many to date. So there's several ways you can get, or you can try to get probiotics in the hive. One is through something called sucrose syrup solution. And this is simply, how you give hummingbirds, a
mm,
hummingbird feeder, and you fill it up with sugar water. It's
mm,
essentially the same thing. A common practice in beekeeping is the use of sugar water to feed bees and kind of get them going. It gives them a little bit
mm,
of energy to go forage and
mm,
everything like that. When you put microbes in there, the idea is that, or probiotics in there, the idea is that the bees will really consume them, and they do. But the sugar solution has extreme osmotic stress that causes the bacterial cells to lyse. So you're putting in these good microbes into the sugar solution. The
mm.
osmotic stress is causing bacterial cell rupture, and then you're essentially supplementing dead microbes to the bees. You're not supplementing viable probiotics.
Okay.
So that's the major problem that I've been trying to kind of, I spread awareness on,
Mm-hmm.
and also trying to change the research that's being done on that as well, because you're not getting an actual true answer of what the effect of delivering viable bacteria to the bees are. And that's not
Mm-hmm.
to say that that can't have a beneficial effect, because it might, right? It might have
Mm-hmm.
a postbiotic effect.
Sure.
The
Okay.
postbiotics are
Yeah.
becoming a thing. Um, the way that another way of delivering probiotics to the hive is through something called a pollen patty, and this is essentially a supplement that beekeepers give to their colonies to ensure
Mm-hmm.
nutritional adequacy. It's like a supplement, right? It's
Yeah.
protein powder essentially, but based on pollen substitutes.
Yeah. Yeah.
And
Yeah.
then what you can do is essentially mix the probiotic cells into that,
Mm-hmm.
and the bees take it up. This method is much better. It has much higher viability, or maintains much higher viability of the probiotic cells. It's good, but it also relies on the bees orally uptaking and then dispersing it throughout their hive. Another way that we were testing, or that we tested in these studies, were simply by spraying the bacterial cells or the probiotic cells all throughout the hive. And
Similar
you,
to like, I guess, the dusting of antibiotics or something like that.
yeah, so yeah, similar to that. And I mean, another approach could very well be to dust freeze-dried bacteria on the hives,
Mm,
right? But it's just a little bit harder for distribution-wise.
right.
We just essentially tried an isotonic solution, which was phosphate buffered saline, and it keeps the probiotic cells very viable and kind of ready to go, right? And we just sprayed all of the frames, and we showed that that method was highly effective at reducing certain pathogens that are found in the frames of the honeybee hives. And it could, over the course of 24 weeks, it almost completely reduced an important fungal pathogen. And one thing we noted though is that delivering the exact same probiotics in a pollen patty
Yeah.
or a spray, which we called the biospray or biospray, or sorry, biopatty or biospray,
Mm-hmm.
had different effects.
Mm,
So you can have the exact same probiotics delivered in different vehicles,
yeah.
and the effect or on the health of the bee is different. And this was just really kind of a breakthrough finding, because it demonstrates that you have to pay attention to each step of the process, right? You
Mm-hmm.
can't,
Mm-hmm.
not only does strain type important, the strain type of probiotics is crucially important to their function, but also how they're delivered. You can think of this, and you can relate this back to humans with the development of probiotic capsules that
Yeah.
don't actually degrade until they get deeper into your
Those enteric probiotics or whatever. Yeah.
yeah, exactly. And
Yeah.
there's a lot of evidence to support that they do have they can have more beneficial functions if those probiotics are released deeper into your gut, as
Mm-hmm.
opposed to just being ate up by your stomach acid or whatever else. And
So
so
what would the takeaway be for a beekeeper who's watching this then, like in terms of I know it's a bit too early, but I'm sure people are probably anxious to maybe try these sort of things. But like you said, like the delivery method is really crucial in terms of, it could have different health benefits or such. Like, is it important for beekeepers to understand sort of what the goal is, I guess, in mind? Or yeah, any advice for beekeepers on this?
yeah, I'd say that there's probiotics that beekeepers can buy now, but they're not validated at the strain level
Yeah.
for the most part. But if they're going to use them, because there's who knows, there are maybe some benefits and people are going to use them. I would say to try to ensure the greatest chance of success, deliver them through a pollen patty, through
Mm,
mixing them in like a physical matrix or a solid matrix, as
mm,
opposed to using them in the sugar water, because you're
yeah.
just killing the cells that way. That would be
Yeah.
my advice at current.
Yeah.
um, yeah.
Cool.
And also
Uh,
to sign up for the Canadian Bee Gut Project, so we
yeah.
can
Yeah. Yeah.
help monitor their microbes. That would be another one.
Yeah, for sure. I'll definitely include that site in the link, that site in the description for sure. Any major other takeaways for you in terms of your research or things that you want to share with folks?
um just kind of to spread awareness of the Canadian Bee Gut Project, and to kind of stay focused on bee microbes, and think of things you know as deeply and as thoroughly as you can. It's not always as simple as saying a probiotic is good. It's like, why is it good? It's good for a
Yeah.
certain function colonization isn't, you don't always need colonization to have a benefit, etc.
Yeah, cool. Hopefully you got the 2 questions I send every guest. Did you get that? The 5
Yeah,
for dinner. Did you? Okay, good. I just don't want to surprise you. If you didn't get a chance to think about it, then we could totally skip them. But if you're good, do you want to end off on those 2 questions?
Yeah, sure. I thought that was those are really fun questions. For the dinner, 5 people dead or alive, I'd want to
Yeah.
have dinner with. So Hippocrates would probably be the first, I would say.
Okay. Okay.
All he's famous for the statement, all disease begins in the gut, right? I think more and more
Yeah.
research is kind of supporting that too. And he was just such a fan. I mean, I took a history course, and a large component of it was on Hippocrates, and I think his entire life is just so fascinating.
Yeah.
It's like he was just so ahead of his time, over thousands of years ago, and it's like he knew more in some ways than we know now. And also because he estimated age of 80 to over 100, kind of defies the general perspective that human lifespan was historically much lower than it is now, right?
Yeah,
But we don't
for
know
sure.
that that's yeah. So it'd be cool. It'd be really cool to have a glass of wine with him and hear what he had to say.
I think you're the only
um,
person so far out of all my guests who said Hippocrates. So
oh,
yeah, that's
all
cool.
right.
Yeah,
I'm surprised. He's great.
I know. I agree. Yeah.
Everyone's first answer is probably like Albert Einstein, eh? Is that all?
I don't think I've gotten a lot of Einsteins.
No.
Yeah, no, I think the most common one has probably been like a religious figure like Jesus or something like that.
Oh,
But yeah,
yeah.
anyway.
I
Yeah.
mean, that would be cool. The second one would be Dr. Andrew Cutler, which is
Okay.
he developed the low dose chelation therapy for removing mercury from your body. Actually, we talked about mercury chelation and
Yeah.
probiotics and things like that. The reason I got into research was actually I had got mercury poisoning from eating fish when I was younger.
Oh,
So it's
okay.
yeah. So
Okay.
I had studied a lot of his work, and
Yeah.
he had developed these therapies and things in the early 2000s, and when we didn't know much about the microbiome, I would love to
Mm.
discuss his understanding of how the microbiome fit into heavy metal toxicity now, current day. I just
Yeah.
think it would be a fascinating conversation. Next would
Does
be
chelation chelation therapy actually work? Do you know?
well, yeah. What do you mean? And what do you
Like,
mean? What
because
do you mean? Does it work?
well, I don't know. I don't know enough about it, but I think the idea behind chelation therapy is the removal of heavy metals and toxins. Am I wrong about that, or?
Yeah. So chelation therapy is referring specifically to the removal of heavy metals in general.
Okay. Okay.
And yeah, I mean it does work. There's tons of clinical evidence on it, and there's the way that most of the time it's done is either intravenously or orally, taking basically a compound that has a high affinity for a heavy metal of interest, and it
Mm-hmm.
pulls it out of the body either through your urine or through your stool.
Mm.
Um, the issue with the current treatments, or the current mainstream medical advice, is that they're
Yeah.
using really high levels of these chelation agents, so they pull out tons of all of the heavy metals, right? And then it can't hold on to them all before it gets out of your body. So what happens is it's taking all of your cells, and then it releases some of it, gets some, it obviously removes some of it, but then the remainder is free in your bloodstream
Okay.
and has really, it can have really high reactive oxygen species damage and things like that. Just kind of disrupting it, taking out your cells, putting it back in your cells, and
Mm-hmm.
it can have really poor consequences on your health.
Yeah.
So the Cutler method is basically just really, you're taking a lower dose of the chelation agent more
Mm-hmm.
frequently,
Mm-hmm.
to remove it. So I did it it took me 3 years to get rid of mercury from my body. I had to
Okay.
go get myself tested many times through a urine challenge test, and I took DMSA. I was going to say DMSO. I've been in the lab for too long. DMSA, like every 3 hours for 3 days of the week, even during nighttime. So it works, just takes a long time, and it's
Yeah.
yeah, it's I think obviously I had some sort of sensitivity to absorb more mercury than the average
Mm-hmm.
person, but it was an experience for sure, and in
Yeah.
my opinion, it definitely worked.
Awesome. Number
The
3.
third one The third one would be Weston Price, Dr. Weston Price. He's a dentist known for his theories on nutrition, physical health, and yeah, in the early 20th century, he studied various diets of cultures and tried to correlate them with dental health and overall health, leading to controversial conclusions about processed food and nutrition. And one of his things was how certain cultures, the consumption of foods with high vitamin K2 content led to proper bone development and also straight teeth. And just really, he had a lot of really cool, fascinating findings and research done during his time. And I know a lot of it is controversial, but I would love to talk to him. Love to have dinner with him.
Yeah.
Another one would be Steve Irwin, aka the crocodile hunter.
Yeah.
I think just his grasp on animal life and ability to interact with so many different animals was truly phenomenal. He just seems like a great guy too. Yeah. And finally, I'd say Arnold Schwarzenegger would be a good one, just to hear him talk for half an hour over dinner.
Oh, that's
Try
cool.
to give
kind of a. I feel like that was a bit of a random one that you threw in there.
Yeah. Well, it was just on my last one. I remember when I was in Los Angeles, I walked by Gold's Gym because apparently, he's a spot there, and I was always wanting to see him there. So it kind of just, when I was thinking of people I would like to have dinner with, I was like, well, I tried there.
Yeah. Have you ever been to Muscle Beach on like yeah, it's a pretty cool place
on the strip there. It is really cool. I mean, it's it's so interesting. It's a different like form of life, I feel like, in that region.
It absolutely is. Yeah. Okay, last question. Besides the circle of life, what do you know for sure?
Oh, this one I was actually thinking on my walk earlier. It's like, what do you know? You know that you die. That is the circle of life. I would say that microbes. I'd say that microbes impact all stages of the circle of life, and also impact my macroscopic reality as we know it. That's what I know.
That's awesome. Brendan, I gotta say, I love this field. I feel like I could have this podcast just dedicated to the microbiome, because having you on, having Emma on, having David on, it's just been really a pleasure to kind of see the microbiome from different perspectives. And this conversation in particular has been really neat. There's some technical pieces that I'm gonna have to revisit and listen to again that I think just maybe went by my head, but it's just really interesting to understand how impactful the microbiome can have on the bee health. We talked about potential interactions with plant health, the similarities with humans, and the takeaways there. And then we had that front end conversation about probiotics, which to me was a bit mind blowing, because I kind of walked away, like I said, thinking that probiotics might not be might not really have any benefits, but that sort of really distinction between if you're not looking to colonize, there are actually still some benefits there. So that was really neat to talk to you about. So yeah, just really appreciate the conversation, and your work sounds super cool. If you're ever interested in coming back in the future, always happy to have you, because there sounds like there's so much work that's going to come out of your research. So just like best wishes to you on the Canadian Bee Gut Project and all your research, and thanks for making time for me today.
Thanks for having me. It's been really great.
Awesome. Thanks so much, everyone, for joining in. Like, subscribe, do those wonderful things, and I'll see you in the next one. Cheers.